second clap.
That was hardly a first. Professor Uncle, take one.
All right, fantastic. No, I don't know what to ask. How are you?
I'm well. How are you, Stuart? Long day, eh?
Long day. Long day's journey into night.
Well, now you're in Adelaide, you'll be revived. It's the city of excitement.
Your city's just too far. It's just too much. You enjoy Adelaide?
Very much so. I've been here 40 years or so. My
How'd you
family
end
is
up in
here.
Adelaide?
Well, I before Adelaide, I was at the Walter and Eliza Hall in Melbourne. And they offered me a very good job here. So this is 85. And I've been here ever since. I haven't regretted it for a moment.
Wow, and before then?
I was
Okay,
in
yeah,
London,
yeah,
London,
okay,
London for
Now-
4 years.
How did you get involved in science? When and how and why?
Well, it's when I look back, it's it's difficult to pinpoint a time, but you know the profession gets mixed with your personal life in many ways. So I did medicine in Rosario, in Argentina. Finished medicine, and then the country was going through serious turmoil. It was basically a civil war developing, and as a young doctor, I thought maybe I would be better off somewhere else. So with a friend of mine, we went to Brazil, and we worked in Rio de Janeiro, for a couple of years. And I was very lucky because I met an Australian who had been working in London and Cambridge in the Medical Research Council. I was on a sabbatical for 1 year. And surprisingly, he was working there, but there were nobody around him that could learn from his knowledge, his experience. And because I spoke a bit of English, they took me on. And I worked a couple of years in Rio, and then I went to do a PhD in London with this amazing scientist called Colin Sanderson.
What did he do?
Well, he's passed away now, but he was a remarkable cell biologist. He was strong in time-lapse cinematography to actually see how lymphocytes, white blood cells, recognized and attacked cancer cells and killed them in a petri dish. And he studied that mechanism of recognition, engagement, and killing. But when he was in Brazil, because in Brazil, like in Argentina, there are some parasitic diseases, he worked on Trypanosoma cruzi disease, which is a parasite that invades cells in the body, in the gut, sometimes in the heart, and causes serious problems. It's called Chagas disease, the disease. But interesting, the cells that are the most effective at fighting this parasite are not so much T cells, but other type of white blood cells called eosinophils. So my first scientific paper I wrote with him was on eosinophils. And that, I think, going back to the original question, that triggered my interest in these cells, the rest of the family of white blood cells, called myeloid cells in general. And ever since then, I've been just fascinated by what they do, the good things they do, how they protect us, but also when things go wrong and cause chronic inflammation. So I've been stuck in this wonderful path ever since.
So you started as a clinician.
I was a medical. Yes, I did see patients for about a year. A doctor. Yes.
Okay, and then somehow you were introduced to research.
Yes.
Okay. Why?
Yeah.
And you took to it. You enjoyed it. What do you enjoy about research?
I think what I enjoyed the most was the puzzle, the unknown, and probably appreciating that if I could solve that puzzle, that would be a unique discovery that could lead to a new way of seeing other problems. So like a stepping stone into the unknown, if you like, but at the same time gathering expertise, knowledge, and arming myself with new knowledge to ask more complex questions.
Have you answered any complex questions?
Well, we're continuously answering questions. Basically, how the body works, how the body functions at different levels, how the cells respond to the immediate environment, whether the cells are in the bone marrow or in the liver or in the spleen. And what regulates how cells behave is what interested me for the good part of 40 years. So, blood cells are produced at a specific rate, so many per day, and so many are discarded on that same day. So that's a process that is tightly regulated, but also is subject to modulation. So if you lose a lot of blood and red cells have to be remade and remade quickly, then the body releases some hormones that instructs the bone marrow to now produce, more red cells, more blood cells. So it's a wonderful process that we view that as in two spheres, if you like. One steady state, what happens every day to you and me when nothing goes wrong. But the other one is of emergency situations where, as I said, there is an accident, you lose a lot of blood, and you need to get that blood back, regenerate that component. So all of that is tightly regulated by small proteins, peptides in the blood called hormones or growth factors. So I've been studying this interaction between these growth factors and cells in the body for a long time.
What's cancer? I've asked 22 people. Very similar and very different answers. And so I'll ask you because everybody asks you probably. What is cancer?
Well, I think I just mentioned how normal processes are tightly regulated. The cell responds to the environment and signals inside itself in a specific way, well regulated. Cancer is really the subversion of that otherwise normal signaling. So these instructions go wrong because it's a mutation in the cell, or there is too much stimulation of the cell, or the genes in those cells have changed how they come together, and now the cell responds in a very abnormal way, is escape normal regulation. To me, that's cancer.
Okay.
Has anybody said something similar?
Well, somebody said we've cured it, at least that one. Some people said, well, and the question of why you got into science, he said, well, the history line was full. So we went in Cambridge, he crossed the line and went to the line in science. So I thought, well, that's how dedicated. And he's a world leader now, so he nearly did history except the class was full. So I've had very many different answers.
Yes, good.
A lot of confusion. Yeah, like what is cancer? We don't know, and we're still investigating, and which cancer, because there are so many.
Yes, but in general, I think the common denominator is lack of normal regulation. Things that should not be happening, suddenly the cell escapes that regulation and goes, what's the word? Just goes her own way. Rogue, goes rogue, exactly. Yeah.
And they hide too.
Well, they hide because we cannot find them. So it's us. We are not opening our eyes in the right place. But
Do they hide in bone? Do they hide in bones?
and
Some cells?
well, they
lie
go.
dormant.
Yeah, well, I mean, you're touching on something that is very close to my heart, which is maybe touching on the stem cells, the origin of the cancer. And some cancer cells are believed to stay dormant or quiescent, and then somehow they are energized again to divide. to expand and to move to other parts of the body and cause serious problems.
So they lie quiet, go to sleep, and wake up down the track and then create havoc.
Well, it's one way,
Bit too theatrical.
one way to look at it. Yeah, I mean,
Is that the way you look at it?
well, yes, stem cells, we all have normal stem cells, at least for blood cells, and they tend to be quiescent, dormant, until there is an emergency, as I was saying, and then those cells are stimulated, are talked into dividing, and then differentiating those cells that gone missing or lost in a hemorrhage or some blood loss. But with the leukemic stem cell or the cancer stem cells, they're probably quiescent in the same way, except that in them something is already gone wrong. So again, comes to the question of defining cancer, and as I said, cancer is just a lack of proper regulation.
So what's the research closest to your heart now? It's T cells.
And not really. Another type of white blood cells, like T cells, but
Let's start me from the beginning on that. So.
well, the blood cells. You have the red blood cells that carry oxygen, and the white blood cells that protect us pretty much against infections, and we cannot live without them, without either. But amongst the white blood cells, you have lymphocytes, like you mentioned. I'm more interested in the myeloid cells, those eosinophils that first attracted my interest and attention, and the neutrophils and the monocytes, and all those cells in between the stem cell and these mature neutrophil eosinophils monocytes. So I'm interested in that differentiation process, because if differentiation is impaired, is affected, or is blocked, that's when you get leukemia. So in leukemia, you have all these undifferentiated early cells that were destined to become useful differentiated cells, but suddenly they're blocked in the differentiation and they become these useless cells that clog the bone marrow and spill over into the blood. And that's what the problem with leukemia.
We've come a long way with leukemia there in the last 40 years.
That's right. That's right.
I get excited when, having gone, being of an age where I grew up with the space race and anything was achievable, everything was a very optimistic time, even though we had the Cold War and everything else. And I guess you were the Falklands War, did you?
Oh, the Malvinas, like some people call it.
Speaking of that.
It's interesting because I was in 82 and I was in England until 81. So in 82, I was in Melbourne working at the Walton Eliza Hall, as I said. And one Saturday morning, I think it was, I got to buy the paper, as I normally did. And there it was, headlines, Argentina invades Falklands. So it was a good place to be in Melbourne at the time.
Well, funnily enough, I just left Sydney. I was in Texas when that happened.
Right.
And then 2 weeks later, I was in London.
Right,
It was
yes.
a very interesting time in world history. It's always been a bit strange, world history. It's always something's coming up.
That's right, that's
Upset the
right.
apple cart.
Yes.
But going back to leukemia, I still wonder how fast. I mean, everybody says it takes time. Cancer research takes time, sure. But compared with where the last 4,000 years, how far have we come with leukemia research in 40?
Oh, yes, it's remarkable. I think we need to be also careful that leukemia is not a single disease, not a single cancer. And many white blood cells are the root of different leukemias. There are some horrible ones for which they're still not cured, like acute myeloid leukemias, but other leukemias that are being treated very successfully. And some rare leukemias also there. And we are working with one particularly nasty, but fortunately not many people have this leukemia, and they were making great progress. We're making good antibodies that could be used in the future as potential therapeutics.
Is I can't. Okay, how do I phrase this? Leukemia is simply a poisoning of the blood.
No, it's not poisoning. It's the bone marrow producing the wrong cells or producing cells that are cut at the knees, so they cannot walk and do useful things. They are just blocked in the differentiation to be the good guys. So in a way, it's poisoning of the blood in that the blood ends up being full of useless cells, preventing the good cells coming up.
And there's hundreds of different types of leukemia.
I couldn't say hundreds, but if you have a lymphocyte and a B cell, a B lymphocyte or a T lymphocyte, then you have B cell leukemia, T cell leukemia. You have myeloid cells, you have myeloid leukemia, you have acute, you have chronic. So there are different types, and they all show different signaling mistakes, different defects in how the cell managed to suddenly not be regulated properly. So the challenge for us is to identify the Achilles heel of this leukemia, which was the signaling mistake, where was it made, and try to tackle that with new drugs.
So that's the key. That's going to
Discovery what the mistake is. It's like taking the car to the MOT and to the garage and see what's the problem is not going well. Sometimes you can point to the brakes, sometimes to the steering. So you got a real idea, but until you go and find what the problem is, you can't fix it.
That's a good analogy. And you've say 40 years in Australia. I'm still adapting to that. And you've ended up in Melbourne, just Melbourne and Adelaide.
Yes, Melbourne for 4 years at the Walton Eliza Hall, and then at here in Adelaide.
So how'd you were invited to start a team here, right?
Yes, well, I came to Melbourne as a young postdoc with Matthew Vadas, who has always been involved with the Australian Cancer Research Foundation as well. He had moved from the US back to Melbourne. And I worked with him and close to another very good team at the Walton Eliza Hall, headed by Don Metcalf, which I'm sure you know very well, and very good people working with him, Nick Nicola and others. And then after 4 years, Matthew Vadas got a very good opportunity here in Adelaide. I was part of his team. It was a good opportunity for me and others. So we came as a group and started a division of human immunology at the IMVS, and then that grew very, very quickly, very well. Very good people came to join us, and that created the Hanson Center for Cancer Research. So, well, it's how can I put it? I think it didn't develop, stalled. Stalled is the right word. But many groups within there remained in different institutions, continuing to doing very good work, I think.
Because we spoke about soccer and teams, and we did. A lot of people brought up sporting analogies. Okay, look, basics here. It's leukemia. You're looking at leukemia. That's your primary drive at the research into leukemia, with a specific leukemia involved.
Well, that's one of the projects,
Yeah.
but really over the years, you amass certain knowledge and insights that point you to one or another disease. So just like those white cells are blocked in the differentiation, don't become useful, something goes wrong in them. Other things go wrong. The cells that already are differentiated fully are supposed to be good, but becomes hyperactivated. So in other diseases such as asthma, chronic inflammation, these myeloid cells go crazy. They're hyperactivated. They're hard to stop for other reasons to the leukemia. But because of the knowledge we have accumulated about how blood cells are regulated, we can envisage scenarios that can be useful for leukemia or for controlling hyperinflammation, such as in asthma.
Okay. Do you want to walk me through just what you're looking at now and how?
Yes,
Is
but
that too much to ask?
no, but I have to also take you back to my days in London doing my PhD, because not far from where I was in Harrow, North London, Cambridge is not far off, and there was a brilliant Argentinian scientist called Cesar Milstein, working with a brilliant German scientist called Koller. And those two people, out of curiosity, they were not set to study cancer or inflammation or anything, but they just wanted to see how B cells, those lymphocytes that make antibodies, how could they be made to last longer or to live forever? And they managed to do that by joining a B cell that makes one type of antibody with a B cell leukemia that continues to grow. And by fusing those two cells into one, suddenly you have like a tumor cell that will grow and live forever, making the antibody that was destined to make. So this is really at the time, it was a revolutionary finding because it's the origin of monoclonal antibodies. And these monoclonal antibodies are now one of the most common therapeutics used in cancer and other diseases. So to me, it was inspirational because not only I was physically close to a lab that was at the forefront of these amazing discoveries, but also taught me the power of curiosity. Just trying to do something well, have a good question, and then the applications of these findings is a secondary thing, very useful, it has to happen, but you need first to have the breakthrough, and then you apply it in many disease situations. So now monoclonal antibodies are applied to many, many diseases. But that lived with me in terms of driven by curiosity, study something important, and then look at the application later on. So for leukemia, for asthma, we have programs that try to develop monoclonal antibodies that can control this leukemia or kill the leukemic cells, antibodies that can switch off these myeloid cells that are hyperactivated, block them, control them. So it's like two things coming together, the insights into the signaling, what goes wrong, and secondly, developing with this technology of monoclonal antibodies, they've been around for many years now, but it's perfected ever since, how to use that in the best possible way to tackle disease.
Have you had any breakthroughs in your life or been part of any?
I'd like to think so. Understanding again how cells work is basic. Is what keeps many of us awake at night. And maybe this is a time to bring up the question of collaboration because we've done pretty well, I think, and develop a very good team. We have very bright people, but sometimes, you cannot do everything. So I've been lucky to develop some very good collaborations over the years. And one that springs to mind is a 25-year-old collaboration with Michael Parker at Bio21 in Melbourne. He's famous crystallographer, a famous, well-recognized structural biologist. So he can look at proteins with exquisite detail in a three-dimensional way. And having that very, very close detailed look at these proteins gives you, again, extra clues as to what we are doing from the point of view of the biology, how cells are responding. So you ask about the breakthrough. One breakthrough we had a few years ago is we couldn't understand how a cell with a receptor, with a protein on the surface that could recognize a hormone, it was signaling because it just didn't make sense. It was impossible for it to get activated. The types of the receptors were too far apart. But we purified the proteins. Team Hercus was fantastic.
Sorry, you have to go back and explain that a little bit more. The path, the distance between the receptors would not activate. So just.
Well, we figure out that the receptor was activated, but the way the receptor looked, just by looking at the sequence, at the one amino acid after the other, suggested that the important parts of the receptor that should trigger the cell to do something were not in the right position. So that didn't make sense to us. So we sought to purify the protein and get an idea of what it looked like, the real structure, not just reading all the building blocks what's after the other, because that's a code. But how it all comes together, we don't know. Only through structural biology, crystallography, like Marco Parker does, we can get to know if this building block that is another one 20 blocks away, are they in a three-dimensional structure, are they really next to each other or are they far apart? And that's what his technology allowed us to do. So with that technology, we discovered that, in fact, the receptors on the cell, they were too far apart in this sense, but because there was another receptor behind, now these were very, very close to each other. And now these two explain all the signaling that went through. So it was a beautiful piece of science where the biology and the structure of these proteins came together beautifully.
And then what? It came together. So that was the aha moment for Michael, anyway.
Well, it was a bit of a little eureka, because receptors. There are many types of receptors, and this was a new, if you like, a new paradigm, a new way to show how some receptors activate the cell, trigger tickle the cell, that was not suspected of before. So immediately that visualized to us how the cell became activated and gave us new means to maybe interfere with that process when we wanted it.
And that's what you're doing now? You're still carrying that on?
We are continuing that in the sense of studying similar receptors that play a role in asthma to see if there is some nuances, some differences that provide a weakness for us to attack, to control the activation of that receptor on eosinophils. But the other thing that did was to then, we had to resort to another technology. And this is some in vivo work in mice, and also genomics technology. And this collaboration was with John Dick, a very renowned stem cell biologist in Canada. And with him and Michael Parker, the three labs together, we managed to show that this receptor on the cell depends how the different parts of the receptor are expressed, affects stem cell behavior. So the stem cells will respond one way by staying quiet and remaining stem cells, or if there's more of this type of receptor exposed, the cell becomes more of a mature cell and becomes differentiated. And because stem cells are at the root of many cancers, well, especially leukemia, I think we got a handle now in how to interfere with leukemic stem cells.
They brought you to T cells.
We don't work very much on T cells, but what we are doing, again, a collaboration with a colleague of mine here, Marco Brown, at the Royal Adelaide Hospital, is the antibodies that we have made, they can also be grafted onto some T cells. So those T cells now become very clever at recognizing what the antibodies recognize, the leukemic cells, and we're planning to use them to tackle leukemia.
Wow. I mean, does AI help you much? Is AI what would the world look like without AI? I suppose.
Well, AI, I think, is another fantastic tool, which essentially this power, computer power. That's what it brings. The big advances in the last couple of years is the computer power has enabled artificial intelligence to be much more efficient. So especially when it comes to genomics technology and examining the role of many genes in many diseases, that will make a big difference for diagnostic and for prediction, and maybe in the future to finding therapeutics that interfere with the new discoveries that we made in how the genes talk to each other.
Making me think. So, what work me through? What experiments are you doing at the moment? What are you hoping to find out? What's your aim?
Well, one of
Is that
the
too broad, or is it?
no, I mean it's exciting because you come to the lab every day and you want to see results. So you have to think of what is the next experiment. And one experiment, is done by Tim Hercus that you met earlier today, is to see how an antibody that we made against this receptor called the IL-5 receptor, how it recognizes it, and make a complex, and then in collaboration with Marco Parker, see if that antibody is useful, a potential useful new drug to see how IL-5 stimulate eosinophils, which are the problem in asthma. So that's one avenue. The other experiments that are really looking interesting is the rare leukemia that expresses a mutation. Is a mutation of a protein that now has something else added to it that is not normal. And we made an antibody to that extra bit. And this is another interesting lesson for me because this was done by a postdoc in my lab called Denis Vorogov. And he wanted to make an antibody to that extra bit sticking out of this protein, the abnormal bit. And I was against it. I thought that's not going to work because you need to have a look at the 3-dimension, everything. Let's sort out the structure first. And he didn't listen to me and went ahead and made an antibody that sees that extra peptide. But to my surprise and my delight, I recognize it's also, the whole wrong mutant protein. So suddenly that antibody becomes a potential therapeutic, a very useful new drug to that type of leukemia. So it shows that sometimes you need to give young people the opportunity to follow a hunch and to challenge you. And, I'm delighted when they get it right and I get it wrong.
Can research ever be called a failure? If the experiment is correct, it's just one more thing that is not doing something. Is it just as useful?
Oh, very, very useful. I think in terms of discovery, what you try to do is disprove something more than proving. You try to disprove by any means you can. You try to find reasons why, this is wrong, and you cannot disprove it. Well, this looks pretty good. So failure, could be seen as disproving something. Okay, I was wrong. Okay, good. Quickly close that road, stop, and move to the other things that you suspect could be at work. So I think an experiment that gives you a negative result could be incredibly insightful.
Yeah, negative is not failure. Negative is
That's
just
right. That's a good way to put it.
It was Edison. He said, "I haven't failed. I've just found another way of how not to make a light bulb." So it's like,
That's right.
and you get to a couple of hundred. Yeah, then we got it now. I mean, Are you excited when you come to work? Yeah, every day it's like,
Yeah, absolutely, absolutely. Uh, Always thinking of you have to repeat something, obviously, but then what is the next experiment? And it's like sometimes my analogy is like going up a staircase, a spiral with windows, and then every step, you get to a window, and it's a new way of looking at things. You get a bigger picture of what's going on. So some is one step at a time, but suddenly, you get to the roof. And then that's the big breakthrough or the big paradigm shift. Suddenly you can see very clearly what was not possible before.
and quite often there are breakthroughs that people don't think much of until 20 or 30 years later and go, that works with, because it's a big jigsaw puzzle in some ways, isn't it?
That's right. Yeah, no, it is. It, in a way, it's a jigsaw puzzle. But also the other analogy is like, being an explorer in the 16th century, where you think you're going to a new place, but with the science at the time, you're not going blindly. You think you're going somewhere in general, but you don't have the tools that you have today. So you set off, and you may end up in the wrong place with a lot of uncertainty. So sometimes what we do is a lot of uncertainty, and it's sometimes finding a result that doesn't make sense, but you need to analyze it, and eventually it can make sense.
That's a good analogy. I like that one. Your experience with the Australian Cancer Research Foundation, do you want to just so they're happy?
Oh yes, no, I think it's a fantastic foundation. Well, I think it was set up for all the right reasons, that help in a meaningful way the cancer research effort in Australia. Secondly, I think it was very clever the way that was designed to help with infrastructure. A lot of the grants we write for support as for people, as for consumables, which is very good, but equipment and new technology is absolutely crucial. So if I can digress for a moment, I think for what we do, there are three things that for me are super important. One is people, having the right people, the quirky, lateral thinkers, intelligent, hardworking, and so on. Two is technology. New technologies make a huge, huge difference. Remember the Helicobacter pylori story, that causes ulcers, causes ulcers.
We have to go back. I was fighting that. Been fighting that for a while.
No, that's right.
Go back to the story, the Flory story.
Technology is absolutely essential. New technology offers new opportunities. About 40 years ago, there was a new instrument was developed, was the flexible endoscope. Allowed gastroenterologists to get really into the stomach and take biopsies and then analyze them. And thanks to that technological advance, Helicobacter pylori was found, the causative agent of duodenal ulcers, and it's a big risk factor for gastric cancers, adenocarcinomas. But a key element in that was not only the physician and the pathologist that observed the growth of the bacteria and then did all the required tests, but the fact that there was a new instrument that allowed biopsies of the stomach to be taken out and then analyzed. So technology is absolutely critical. And
You take it for granted sometimes when it's been around for a while, but you forget.
That's right.
What a big
Yes.
breakthrough it
Yes.
was.
Yes. So this is Robin Warren and Barry Marshall, and Robin Warren was a pathologist. In fact, he was born in North Adelaide, just across the river there, and he was able to look at those biopsies and observe the bacteria, and then with Barry Marshall did a lot of good studies to show that that was the cause of duodenal ulcers. So again, the equipment and the technology is crucial. And the third thing that very few people talk about is having very good biobanks. So these are repositories of human specimens. We have a fantastic blood bank here, for example, in South Australia, where blood is being collected for many, many years, more than 40 years, from many patients. When the leukemia arose, after treatment, when the leukemia came back, and all those samples from different patients stored away are an absolute gold mine to understand when things go wrong, when things can be fixed, when things go wrong again. And these provide amazing tools for the future to understand the basis of these diseases and arrive to new cures.
Wow. I mean, I sort of understand. It's like
Well, because
it's a library, it's a blood bank, it's a library.
now you got cells frozen, right? It frozen, and what happens is with all this new technology developing, especially genomics technology, you can analyze thousands of genes at once now, and the costs have come down dramatically. So you can begin to see in those patient samples, because they are sequential, at presentation, diagnosis, after treatment, and then when the patient relapses, how the genomics, the gene landscape, how it changed. So what was lethal versus what wasn't, and what is the new opportunity to come with new drugs. So the new technology, like genomics technology, which is unbelievable, is revolutionizing treatment and diagnosis. But if we have a good biobank with very good clinical history, so the patients are followed over the years, it's absolute gold. And we are very lucky in South Australia, we have that.
So the equivalent I'm thinking of is like when the technology catches up with you.
Yeah.
It's like, well, I think recently they identified Jack the Ripper through DNA.
That's right. That's
So
right.
it's like, now that's
Yeah.
been unsolved, but there you go. So it's the same thing. You don't until you get the breakthrough. Then would you be the equivalent of pushing against the door in unison with all your team, and then everybody falls through to the other side and goes, "Eureka!"
That's right. That's right. Yes. Yes. No, it's amazing resources. So things are very exciting at the moment here. But going back to the cancer, the Australian Cancer Research Foundation, I think the first big donation was to establish the Hanson Center, and that was led by Matthew Vadas. And I think the ACRF had just been founded by Peter Abels. So that was $1 million that came to South Australia to establish the Hanson Center. And you should talk to Matthew about it. He was a great player, and he was, the organizing of that bid to the ACRF, and it was fantastic to then have a new building to do our cancer research. And over the years, we had many donations from the ACRF to establish different facilities. The genomics facility in South Australia was thanks to the ACRF in 2009. They gave us $3.5 million, which then we used that solid foundation to bring more money in to create this facility, which is working today very well in SA pathology. We grew that to $10 million initially. So that was from 3.5 to 10 millions with leverage. So since 2009, we had this beautiful cancer genomics facility. In 2005, we were supported again through a grant to establish an imaging facility, which you've just been to see it actually in the 2 levels below. And
what's it do?
well, this is very, very nice equipment that helps us visualize in 3D the interaction between cancer cells and the immediate microenvironment or surroundings, and how the environment actually is an interplay between the microenvironment and the cancer cells that ultimately can result in these cancer cells being controlled or grow uncontrollably.
Why is that important?
Well, it's important because we have a clear focus on cancer cells, but I think we shouldn't lose sight of what's surrounding them. They're, like in one's life, we have good influences and bad influences. Unfortunately, we don't see the bad influences until it's too late. Well, that's what I tell my kids. But the environment does play a clear role, and depending what is produced there, how the cancer cells may be re-educated or badly educated, that can cause problems.
So with this imaging, you can see it three-dimensionally?
Yes, that's right. And we're using also with new technology again, to probe cancer cells and the microenvironment with 40 different antibodies with different colors that will give us unique patterns and identify rare cancer cells, for example, that are responding in a particular way.
Okay, bearing in mind we've looked at some of the video that's going to come, do you want to try and explain something that would make sense when we're looking at that, without you necessarily being there, but hearing your voice?
Well, I think it's pretty much what I said just now, that biopsies we take from patients with breast cancer, for example. There is a particular type of breast cancer that sometimes can become invasive, and to be able to predict which patients will have an invasive breast cancer. Invasive means it will lead to metastasis and will need much more serious treatment. Predicting which of those patients will develop this invasive phenotype would be very important for designing how they will be treated. And also predicting those that will not become invasive is also important, so they don't become overtreated either. So this is a nice collaboration with pathologists at Royal Adelaide Hospital and SA Pathology, and here as well.
And what's Tim going to show us?
Tim Hercus he loves his chromatography. He's going to show you how he can purify proteins.
Well done. It's my fault. Start without saying what he's going to show us, because we'll just cut to him. The importance of what he does. What's the topic? Chromatography?
Chromatography, to separate proteins based on different properties like size.
Why is chromatography important, and how's it work?
Well, it's important because it separates the different proteins we're interested in. And then with those individual proteins, in collaboration with Michael Parker, we can get to know the three-dimensional structure. Those purified proteins, we can also make our own monoclonal antibodies. So those could become tools for understanding how the proteins work, and potentially as therapeutics, as it's happened to us in the past. So understanding the nitty-gritty of what the protein looks like gives amazing insights into what they do and how to handle them.
How? Staining? You stain them?
No. We can. You will see in the chromatography equipment that you get a profile. The detector will see when the protein comes through at different times, which is reflecting of the size of the protein. And then that protein can be used. What we do is mix it with other proteins, the same family, and see how they interact. Because if we get to know that interface really, really well, we can interfere with that in many ways. So we can short circuit, if you like, that piece of signaling when it's going wrong.
Excellent.
Look, it's, do you know, in fact, at the very beginning, you were sort of talking about when you first got interested in, you were looking at proteins and the way that, in fact, they, interact with, what else? Well, there's also in bloods.
In a single word?
No, just the, Oh, it's in your endocrine system. Hormones and proteins interacting with cancer and the fact that these cells go rogue when the proteins lose their identity or whatever. And I just thought, I've heard a lot about proteins, with everyone talking about cancer, and you were just talking about it then. Is the cure for cancer found in actually determining or understanding completely what proteins do? Is it because we don't understand the way that proteins work that we can't get to the causes of cancer?
Okay, just go and listen to the question.
Yeah, is proteins,
Yeah,
is the study of proteins the essential
Yeah,
question
that,
you're
that,
looking for?
When you answer, look at me.
And the essential question, the mystery of cancer, is the mystery of cancer going to be solved by understanding proteins?
I think it's a major quest. The DNA will tell you, will code for a protein, but then the protein itself can undergo changes that the DNA is not coding for. So in cancer, many proteins are faulty, not because the DNA is faulty, but because they acquired other changes. So I think protein is an important clue, as well as the DNA. I think both go hand in hand. So for example, some proteins undergo changes that make them become spontaneously activated. And that's the last thing you want, because you want regulation. You want to turn things on and off. And if those changes make the protein, like an enzyme, the enzyme is a protein, it becomes super activated, then you need to be able to stop that. So you are right. You need to know as much as you can about proteins and how to handle them properly.
Do we know everything there is to know about proteins?
No, we're a long way away. And partly is what I said before, because those proteins can be modified inside the cell after they've been synthesized according to the instructions of the DNA. The proteins are made in the cell by its own machinery in the cell and can undergo further modifications. You can have some biochemical changes happening in the core, on the outside, on the other side, and those play a role in how the protein is going to function. So all of that is still to come.
All of that. Is there anything you want to say about your journey or the future? Or is there anything you want to say?
Oh, I think I've been very blessed. It's been wonderful to be involved in research, traveling to the unknown and looking for discoveries. It's just fascinating. Maybe I can say a couple of things in terms of sometimes things don't go as predicted. It's not all rosy. That is a discovery every week. Sometimes it takes time and requires special personality sometimes to go through what looks like failures, which are not failures, but they are missteps, or not immediate reward. And I think need people who are not just smart but also resilient and can get over that initial disappointment. The other thing I would say is we need a funding system, and this is not ACRF, but in general a national, international system that provides some stability to scientists. Many researchers have 3 years, 5-year appointments, and time flies. And within 1 year or 2 years, people need to worry about the next job, and these people have families. So I think it would be nice to have not just more funding, but a system that allows stability and continuity, and also allows for when things don't go as predicted. We cannot be producing or discovering a paradigm every year. It just doesn't happen, even to the brightest people. So I think the system has to allow for that, the occasional gap. And but again, being rewarding so that the scientists, we have their back. and they can continue to the discoveries that they, sooner or later they will make.
See, it's the same problem we've got with politicians, right? As soon as they get elected, they're on a big campaign trail.
Well, that's the problem, is the short-term cycle of all these things. I think, you should make an investment in a group, in a scientist like they do in other parts of the world for the next 10 years or 20 years. It's a risk, but I think sometimes we can be so risk averse that we forget that the biggest risk is not to do things properly.
I suppose politicians is not a great example because even if they're in opposition, they still get paid.
I won't go there, but
Well, no, if scientists don't get a grant, they don't get paid, and everything stops. It comes to a grinding stop, doesn't it?
that's right. So, there has to be measures where you can even put some money aside for a rainy day. At the moment, the system is very cutthroat. Only 10% of grants get funded by the NHMRC, the IDEAS grant. Only 10% of those people will have 3 or 5 more years. What happens to the rest? The people you have seen, are going to see in my lab, they have been with me for 30, sometimes more than 30 years. And the reason is that, we've been lucky with the success of the grants, but we also have developed a system where we have backups and we have continuity. So Tim Herkus has been with me for more than 30 years. And instead of developing a personality of being of comfort, or the opposite, is giving him peace of mind to think about science all the time and get grants and good publications, good breakthroughs. So I think the scientific community has to make a stronger case for stability and long-term planning, long-term support.
You can't think about science if you wonder where the rent's coming from.
That's right. Well, that's right. You got a family. You have to worry about your kids' education. You have to put bread on the table. So those are the top priorities. But just take some chances. Identify the best groups in Australia, at least, and give them 10, 20 years support. Maybe that's the next challenge for the ACRF. Have a big pot of money for, initially 1 or 2 or 3 big groups in the country.
Put them on speed dials.
Yeah.
I think I've really had it. Is there anything else you want to say? Or?
No, I think.
While we were downstairs, you said a great thing. You said, the whole search for finding, how to treat cancers is like a mystery. It's like, do you want to just make a brief statement about how you feel it's like a thriller, like a mystery?
Oh, I see. Well, it is a mystery with different episodes, and you can think of a breakthrough as an episode in a big quest, and eventually you'll conquer the disease. I mean, chronic myeloid leukemia is a beautiful example where a few years ago it was a death. It was just no cure, and now most people are treated well and develop a chronic disease, or they are cured. So I think we come a long way. I think you have to be optimistic. 65% of cancers are pretty much controlled or cured these days. So that's extraordinary, and we're making big advances. So I'm very optimistic that we're going to improve the health of people very rapidly. But it's exciting as well as a puzzle solving.
That was a great quote. Andrew, do you have anything you particularly interested in?
No.
Oh,
I've been trying to think of a question since Stu.
Steve's getting obscure though.
No, I'm
turn
just-
him off in a second.
but I was going to ask and say, with this amount of cure of cancer, like if there's 65% have been cured, does that mean that it's possible to be cured, or does it actually need government intervention and health programs to make that kind of number, that 65%?
Well, in terms of government intervention. Obviously, the more support from the community and the government, the better. I think the more scientists you can support and the longer term support you can give them is a no-brainer. It's going to be incredibly helpful to accelerate, be a catalyst to accelerate arriving to more cures.
But what about, will we need vaccination programs? Will we need that kind of intervention in people's health to maintain that 65% cure of cancer?
Well, I think we need tackling the problem from many angles, including public health. For example, the question about melanoma. Melanoma is, we should be much more proactive, advising people of the risks of UV radiation and to protecting their skin. The risk of smoking as well with lung cancer. So all of that has made a big difference already. But in the case of lung cancer, many lung cancer patients never smoked. So it's something else going on. And I think, providing support for this scientific community, but also the biobanks. I think the biobanks are really underrated, and they are an incredible source of it's a wonderful tool there to then begin to understand in those patients what went wrong.
So is it that in fact that we governments and organisations need to make research about cancer drivers that we have in the environment now and to give the public notification of those so that we can kind of cure the drivers that make cancer?
Well, the environment plays a role, as I said, and I think the government has made wonderful efforts and educating people on the risk of UV radiation in summer especially, and tobacco and alcohol as well. But I think that's it's not enough. I think we need to understand the basis of what's gone wrong in the signaling in those cancer cells, and that can only be achieved with hard-nosed research, investigation with new technologies. But one thing I would say, and is that sometimes scientists are a little bit frustrated, and not so much with politicians, but sometimes organizations like universities, institutes have very, very strong rules, and sometimes it's difficult to collaborate across different institutions. I think everybody is very well intentioned. Compliance is important, but sometimes it's a big burden on the scientific community to tick every box. And sometimes it's not too straightforward to collaborate between institutions because everybody imagines a big breakthrough every time and millions of dollars coming, which is a very rare occasion.
Yeah, I get that. They don't play well with others, some of them.
My son is an accountant, so I have to be careful what I say about accountants.
It's a noble name, the word count is part of it.
I think they go hand in hand,
My
don't they?
mother was an accountant too.
Oh, sorry.
I remember sitting underneath and going, "What's wrong, Mum? I'm looking for 2 cents. Shut up." The books won't balance on Monday.
Yeah.
I'm thinking about cutting. Do you think about cutting?
Yeah, sure. We could cut.
We could cut.
All right.
Would you like to cut?
Let's have a drink.
Yeah.